Let's talk about muscles today. We're done with bones. I'm sure you guys are excited about that. And now we're going to switch gears. We're not going to learn the different muscles of the body today. We're just going to talk about what muscles are and kind of what they do. All right. So a muscle is obviously an organ that shortens. Okay, and that's really all it can do. It shortens, and like muscles that attach to skeletal bones, when it shortens, it generates force on that bone. And if that muscle spans a joint, that bone will move. So basically just think of muscles as organs that shorten, okay? Muscles have a general kind of structure that we'll talk about here. So if we take the bicep, for example, okay, so we take a whole muscle. I'm just going to kind of draw this guy out. This is just like a whole bicep muscle. This is a whole muscle. That muscle is composed of a bunch of little tubes that are all wrapped together to make that one big muscle, okay? So we'll take all these little, all these smaller little tubes that compose this whole muscle. All right. It kind of looks like that. And these little tubes that are inside the whole muscle, they have a name, and I'm just going to kind of draw one out and make it get bigger. It doesn't get bigger, but you know, it's like coming out of the board, right? This guy is called a fascicle. Okay, so a muscle is composed of a bunch of smaller tubes called fascicles. Okay? Now, these fascicles, they're composed of little tubes. Okay? You got all these little tubes inside the fascicle. Kind of look like that. I'll take one of these guys and draw him out. This guy, all the tubes that make up the fascicle, this guy is the muscle fiber. And what's important here is this muscle fiber, that's the level of the individual muscle cell. So when you think about it, like, well, oh, where's the muscle cell? Well, the muscle cell is synonymous with the muscle fiber. And these muscle fibers, which are muscle cells, are these long cells that are multinucleated. Okay, so they have multiple nuclei. And they kind of look like that. There's just like these long cells with multiple nuclei, all right? Muscle fibers are composed of little tubes. All right, these tubes kind of look like this. These little tubes, if I'm going to draw this guy, make him bigger. These little tubes are called myofibrils. No, so we're just getting smaller and smaller and smaller and smaller. It's like tubes within tubes within tubes. These myofibrils, okay, what they're consisted of is they're consisted of this like big grid of all of these tiny little things that get smaller and shorter when our nerves tell them to do so, okay? So if we were to draw this guy is composed of like all these little grids. And I'll tell you what these grids kind of are. So you got all these little grids that go the length, they just fill up this myofibril, and that's really what does the shortening, okay? So think about myofibril as a grid of little shortening units. So each little square is a shortening unit. And the name of each one of those shortening units is called a sarcomere. S-A-R-C-O-M-E-R-E. So I mean, really, we're looking at like tubes within tubes within tubes, right? What level is the level of a single muscle cell? The fiber. Okay, fiber. Good. Okay, now what I'm going to do is I'm going to kind of go into a little bit more detail about what's going on with this grid. All right? This grid is a series of proteins.
What did you write underneath the grid?
I'm sorry?
What did you write underneath the grid?
Oh, it's a shortening unit. Like it's the unit that does the shortening. And it's called a sarcomere. Sarcomere. Okay, so each one of those little grids is called a sarcomere. And I'm going to show you guys exactly what it kind of looks like. Yeah, short. So it's just like the myofibril. Yep. All the tubes that make up a fiber, a muscle fiber, are called myofibrils. And those myofibrils consist of a bunch of sarcomeres, just a big grid of sarcomeres. All right, so this is what that grid really looks like if you zoom in. You'll have some pretty thin blue proteins. They're not really blue in color, but I'm drawing them as blue. Okay? And they kind of look like that, and you'll have blue proteins that kind of go out like this, and you'll have another little area of more blue proteins, kind of look like that. And then, they go out this other side too. These little blue proteins are called actin. Another name for this little, what I'm drawing here in blue, are called the thin filaments. So actin is ACTIN. Another word for that are the thin filaments. In the middle, in between this grid, I'm going to draw a protein in red that's thicker. Okay? And this thicker red protein is called myosin. And if I were just to highlight one of these little sections of this grid that contains the myosin in the middle, that's surrounded by all these thin filaments, the actin filaments, that is what's considered a sarcomere. So if I were to kind of circle this guy, that is what you call a sarcomere. Good. Now, those thick filaments, the myosin, well, let me write that in there. Myosin is also known as thick filament. These thick filaments, they have these like arms that stick out from them. They almost look like a caterpillar or something with just, arms just coming off, legs coming off of every little part. And those arms are reaching out towards the thin filaments. And they almost look like a caterpillar, or maybe even overhead view of a rowboat with a bunch of, rows coming out of the protein. But what's important to visualize here is that I've drawn it kind of as a two-dimensional structure, but these little arms, which are called myosin heads, are coming out of every angle because there's actin kind of surrounding it in all dimensions, even though I've done the two-dimensional slice. Does that make sense? It's like a three-dimensional structure, and you have actin here and here and here, and you have all of these extensions. These extensions are called myosin heads. Myosin heads, and they are reaching out towards the thin filaments, the actin. And this is basically how the muscle shortens. So, when there's available ATP in that sarcomere, and when this myosin head is able to grab onto the actin filament, I'll tell you that it can't always do that, but when it's able to, those myosin heads will reach out, grab that, part of the actin, that thin filament, and then it'll just pull down. Okay? And it'll do it again. It'll release, it'll reach up, it'll grab, and it'll pull down. And all of these little myosin heads are doing that at the same time. And what happens is these guys are pulling that way. These guys over here are pulling that way. What's that going to do to the width of that sarcomere? It's going to shorten it, right? And so every sarcomere, when those myosin heads are exposed to ATP and when they're able to grab on, they're going to start pulling on actin, and it's going to cause that sarcomere to get shorter. Okay? And if all these sarcomeres are doing it at the same time, like if he's doing it, he's doing it, he's doing it, then the whole muscle fiber is going to get shorter. And that's basically how muscle pulls. Yeah?
You might have answered this, but is that one box? Is that one?
Yeah, that's right.
Right here?
That's right. So what I've done is like this is a bunch of sarcomeres.
Right.
And I've just highlighted a little portion of that. You know, so this would be like 1 box, 2 boxes, 3
That's
boxes.
what I was getting at. Okay.
Yeah, that's right.
Okay.
Yeah, each one of these is a sarcomere, and they're all just packed into that myofibril.
Okay. And they're pulling in towards the middle.
They're just pulling in.
Okay.
They're pulling in. It's perfect. And we might as well talk about it here because it kind of makes sense to talk about it here. But when we contract our muscle, okay, let's think about your bicep. Do you think your bicep can generate the most force when your arm is completely extended, when it's like halfway flexed, or when it's like all the way flexed? Yeah. Who thinks it can generate the most force when your arm's like really already flexed big time? All right, how about who thinks you can generate the most force when you're completely extended? How about halfway? All right, well, halfway is the right answer. And this is why, okay? This is exactly why. When your arm is like completely flexed like this sarcomere is already shortened as much as it can, and the ends of the myosin proteins start bumping in to that actin protein on the end. And so there's really like nowhere to go. And so it can't generate that much force, okay? On the flip side of that, let me draw a different kind of simplified picture. When your arm's really stretched out, I'm just going to draw one sarcomere. You'll have the myosin molecule in the middle, and only like 1 or 2 of the heads can reach out and grab the actin. The rest of the heads have nothing to grab onto. You know, the sarcomeres are too far apart. And so you can't really generate that much force. But when you're right in the middle, so when the sarcomere is about this length, you'll optimize the overlap between all those myosin heads and the actin, and you can generate more force because more arms can reach out and grab up to an extent, and then things just can't get much shorter. Okay? So that's why when muscles, you can generate the most force when they're in a kind of an intermediate length. It's all about the micromechanics of these sarcomeres. Okay, good.
So what makes your muscles hurt when you work out for the first time?
Oh, like when you first wake up?
No, work out.
Work out. Okay, so that's a great question. Working out, and this is how you build muscle through working out. What happens is, when you work out and you're not used to it, or even as you're working out hard and you're training really hard, these sarcomeres get overworked and they actually rip apart. Okay, so they get overworked and they become damaged. These guys just completely fall apart. That induces an inflammatory response. It triggers some pain receptors, some nociceptors, and that's why it hurts. Like literally, you wake up and you're sore. Now, to compensate for that, what the body does is like, all right, well, how many sarcomeres we had yesterday is obviously not enough. Let's repair the broken ones and then pack some more into our muscle fibers. Okay? We don't grow more muscle fibers, we just pack more myofibrils and more sarcomeres in each myofibril to make each muscle fiber kind of more capable, right? And so then you build like a bigger, you pack more sarcomeres into each myofibril. Then you go back to the gym and you lift like heavier weights, or you run further, and you damage those, and the body says, well, we need more, right? And so you just keep building up more of these shortening units due to the damage. And what's crazy is you can look at like the histology of a damaged, worked out muscle, and you'll see all these nice, organized kind of grid of sarcomeres, and in the region that was damaged, it'll just be all torn apart. Just like, I mean, it's damage. It's just straight damage. So that's kind of how that works. Now, we're going to go into a little bit more detail about exactly how those myosin heads grab on to the actin filament. So what I'm going to do is I'm going to zoom in on like this little part of the sarcomere. So at the level of one myosin head, grabbing onto that actin molecule. So here we've got that thin filament. And that thin filament, if you zoom in on it, has specific sites where the myosin head can bind onto it. Those are called binding sites. It can't just grab on anywhere. It has to grab onto these like specific little sites called binding sites. Now, we'll start with this myosin head kind of in this relaxed position, okay? So it's just kind of hanging out. The first thing that will happen is that ATP comes along. I'm going to draw ATP in green. ATP comes along and it'll bind right there to that myosin head. And when it binds to that myosin head, that ATP hydrolyzes, okay? And when it hydrolyzes, it splits into what? Like, so when that energy is released from ATP, what does ATP turn into? ADP and P. Okay, so now you've got, I'm just going to draw this same guy under here. Right? And, but now, this myosin head reaches up here after that ATP changes into ADP, and then P hanging on. It's still attached to it, but ATP just turns into ADP and P. Releases that energy, now the myosin head reaches out, okay? And when it reaches out, it's going to bind onto one of these binding sites, right, if it's available. Yeah?
Is it like a lock and key, or is it just like, it's just going to attach, it's like a glue? Like
It's like, think about it, it's just, once it's energized by that ATP hydrolyzing, it's just looking for something to grab onto. And if one of those binding sites are available, which we'll talk about when they are, when they're available, it'll just grab on automatically, okay? So then, this guy will grab onto one of these binding sites. And I'll kind of draw it like that. Then, once it grabs onto that binding site, ADP and P fall off. So this guy falls off. Now, I'm going to draw this guy again. And we'll go over this again because this is actually pretty tricky stuff. Once ADP and P fall off, so it binds on to actin, ADP and P fall off the arm, and that causes the myosin head to return back to its resting position. Okay, just like this. But because it's stuck here, he's going to return back here, and he's going to pull. And now, nobody's attached to it. So if I were to kind of label this again, we've got step 1, step 2, step 3.
Why does it fall off?
You know, it falls off because it's all about charges, right? Remember we talked about chemistry and it's all about charges? Well, when ATP gets hydrolyzed and it turns into ADP and P, it sticks on there, and myosin's looking for something to grab onto. As soon as it grabs on to this little binding site, that rearranges the charges of the whole molecule, and for whatever reason, ADP and P just pop off. Okay? That's kind of complicated. I think the take-home message here is that ATP hydrolyzing causes the myosin head to reach out. It'll bind onto actin if it can. As soon as it binds onto it, ADP and P fall off, and myosin pulls because it's returning to its resting position. Right? Here's the kicker, though. What's that?
Can you say that one more time?
Yeah. So, you start out myosin heads resting. ATP will bind onto it and then hydrolyze, release that energy. That causes the myosin head to reach up and try to bind to actin. If it can bind to actin, it'll bind onto it. That causes the charges to kind of rearrange. ADP and P and phosphorus fall off. When that happens, the myosin head returns back to its resting position. Because it was grabbing on, it pulls. And look, he's moved from here to here. He's moved over. And that's how it pulls. Now here's the kicker. He's going to stay stuck to that binding site until something happens. What do you think that is?
A reversal.
So this is a cycle. We have to go, what's the difference between this and this? ATP. So in order for that guy to let go, a molecule of ATP has to come in and bind to it right here. So if ATP, if that comes in, this guy will pop off. And once that ATP molecule hydrolyzes, what's that myosin head going to do?
Go back to
Reach out. Right? Then he's going to bind on to actin. We've got ADP and P. What's that going to do? Fall off. What's that going to do to the myosin head? He's going to pull. He's going to shorten. Now ADP and P have fallen off. He's stuck there. ATP comes back in. He pops off. It hydrolyzes. He's activated. This is complicated stuff. All right? It really is. I think the take-home message from all this is that ATP is obviously required for this whole system to happen, right? And if ATP stops coming in, then you're stuck right here. And if you think about it, that's why rigor mortis happens, okay? So after death, there's obviously a store of ATP in the muscle tissues, right? And once the muscles kind of have, well, we'll talk about rigor mortis here in a little bit. We don't have all the pieces to this puzzle yet. All right, we'll talk about rigor mortis here in a little bit. But basically, if there's no more ATP, those myosin heads are going to remain bound to the actin molecules. All right, that's tough stuff. Yes.
So, what about certain diseases like muscular dystrophy? What does that do to the muscle?
That's a good question. So I don't know a whole lot about that specifically, but I think that's more of a nervous issue.
Okay.
So one thing that we're going to talk about, and this is really important, is that each muscle fiber has its own dedicated nerve. And the only way that it's going to fire is if it gets an impulse from that nerve, right? And so these muscle fibers are just totally relaxed. The only way skeletal muscle fibers contract is if we tell it a signal. We send it a signal. And if we don't get that signal, then the muscle fibers stay relaxed. Now, I'll have to just get back to you on that, Alexis. I think it's a nervous issue where the muscles aren't being stimulate it, but I could be completely off there. I'll just have to get back to you, yeah.
So this process happens every time we move a muscle?
Every time. And this cycling is like constant, right? So when you tell, like, I don't know, I lift this basket of markers, all those muscle fibers in my bicep are being activated by nerves, and that's telling all of these sarcomeres to shorten. All of those myosin heads are pulling constantly as long as I'm holding this. It's called, that process is called Crossbridge Cycling. Each connection between myosin and actin is called a crossbridge, and they just continue to cycle. They're just continuing to pull and pull and pull. All right. Good. All right. Now we're gonna switch gears just a little bit, and let's see here. I'm just gonna take a picture because I'm trying to upload some of these online. All right. Now we're gonna talk about how the muscle gets activated. All right. I mentioned that it requires a nervous impulse. Well, the first step of this nerve of kind of controlling when the muscle contracts is due to the presence of two proteins. These proteins are called troponin and tropomyosin. Troponin is like a little protein that hangs out right here next to the actin, small, thin filaments, okay? It's connected to a string-like protein called tropomyosin that covers those binding sites perfectly, okay? It covers them. Let me label the names of these guys. So the brown is troponin, Troponin, and the green, that's the string-like protein that covers up those binding sites perfectly. That's called Tropomyosin, right? All right. Under normal conditions, when the muscle's at rest, tropomyosin is covering those binding sites. Do you think there's ATP available in the muscle cell, like just at rest? Yeah, we've got ATP in there. Like all the time, we've got a surplus of it because we need a lot of ATP for when that muscle wants to fire. So if these binding sites are blocked and there's tons of ATP, what are all the myosin heads doing? They're reaching out, they're ready to grab on, but they just can't grab on because all the binding sites are blocked by tropomyosin. Does that make sense? It's like they're ready to go. They're at this stage, but they can't attach because tropomyosin is blocking all those binding sites. Like they're ready to go, but they just can't. And that's happening 24/7. Okay? Now, the kicker is when calcium is exposed to this system. Calcium's, a really important ion. When we send a nerve impulse to our muscles, it causes a sequence of events that ultimately releases calcium all over these sarcomeres. So if I were to take calcium, all these little calcium molecules, what calcium does is calcium binds to troponin, and when calcium binds to troponin, those charges rearrange. Tropomyosin changes position and exposes all those binding sites. All right? Yeah, it's great. This is hardcore physiology. If all those binding sites are exposed, we got plenty of ATP, all those myosin heads are ready to go. What's going to happen?
They're gonna start connecting.
Yeah, they're going to start cross-bridge cycling. Perfect. And it's all because calcium was released into that area. Calcium bound to troponin. When that happened, it caused tropomyosin to move out the way, expose all those binding sites, and the muscle can start shortening. What caused calcium to be released? a nerve. Like that was our nervous signal, okay? And that's how we kind of control when contraction occurs. A nervous signal releases calcium, boom, and then we're good to go. All right? Now we're going to talk about something about the specifics of how that nerve stimulates the muscle. And this is actually good stuff. Good stuff. I'm going to draw the nerve in green. Each nerve fiber, or each muscle fiber has its own dedicated nerve, okay? So here, we haven't really talked about neurons yet, but this is the tail end of the nerve fiber. And here is our good old muscle cell, our muscle fiber that we want to stimulate. So an electrical signal, this nervous impulse, is going to be sent down the length of this nerve, okay? And when it reaches the muscle, it's going to tell that muscle to contract. Now, this interface between the nerve and the muscle, so if this is nerve and this is muscle, it's really a single neuron, and this is a muscle fiber. The junction between those two guys is called the neuromuscular junction. N-E-U-R-O, muscular, and then junction. This is what happens. First step is a electrical signal flies down the nerve, boom. It reaches a dead end though. See, there's this like gap between the nerve and the muscle. It's called a synapse. That's part of the neuromuscular junction. When it reaches this, it tells all these little bubbles that are contained in the end of that neuron or that nerve to release specific little chemicals into the synapse, which is the little space that separates the nerve from the muscle fiber. So it tells all these little bubbles that contain these chemicals to release those chemicals into the synapse. All right, so all these chemicals get released in the synapse and they diffuse right across. Anybody know what those chemicals are called? They're neurotransmitters. What neurotransmitter is so we've got a bunch of neurotransmitters. We've got like dopamine, we've got serotonin, we've got glutamate, but there's one neurotransmitter that's always released by nerves to stimulate muscle cells. Close. That's a hormone. That's close. It's acetylcholine. Okay? So this is a really important neurotransmitter. So I'll just label this guy, and he is acetylcholine. Every nerve that stimulates any muscle in our body releases acetylcholine onto that muscle. It's just a specific little chemical. Now, the muscle fiber itself is, and the abbreviation for acetylcholine is ACH, capital ACH. Now the muscle fiber is actually covered by its own little version of like nervous type tissue. Okay, so imagine it's like it's covered by this sheath of, nervous tissue, and I'll draw that in purple. So surrounding the whole muscle fiber is this like big sheath of nervous tissue that like surrounds all of it. All right. That sheath of nervous tissue is called the sarcolemma. And when the sarcolemma has little receptors that bind on acetylcholine, and it causes an electrical signal to be to be stimulated in the sarcolemma, and that electrical signal will fly around the muscle in all directions. So if the nerve stimulates the muscle like halfway down, that muscle fiber, The sarcolemma, just think of it as like a secondary nervous pathway, and that signal just shoots all around the muscle. Okay? I'm going to show you the figure in the book here because my drawings always get really bad at this point. But that signal from the sarcolemma dives deep into the muscle fiber, deep into all the myofibrils that I drew, and all the sarcolemma, through a special little pathway called the T-tubule. And once it gets in there, it causes special little containers of calcium called the Sarcoplasmic Reticulum to release that calcium into the sarcomeres. Once the calcium gets released into the sarcomeres, what happens? Activates crossbridge cycling. Yeah. Here, I'm going to show you guys, because I've tried to draw this so many different times and it never really works out. So I'm going to show you guys, I'm going to direct you to a nice figure in the book that that kind of summarizes this really, really well. Oh, and yeah, this is just a really nice figure, and it's basically the same thing that we just drew. But the key point here, like if you're going to look at a take-home message of this, what stimulates the muscle fiber? The nerve, right? A nervous signal that releases neurotransmitters in the form of which neurotransmitter gets released? Acetylcholine. That stimulates the sarcolemma, which is just a big kind of nervous thing, nervous tissue that surrounds the whole muscle, and that is what kind of kickstarts this process. Do you have a question? Yeah, so the take-home message is that a muscle fiber is stimulated by an electrical impulse from a nerve, right? When it reaches the muscle, which is the neuromuscular junction, acetylcholine gets sprinkled all over that muscle, and that stimulates the sarcolemma, which is nervous tissue that surrounds the whole muscle itself. Here, and this is perfect. This is the same topic, and I'm just gonna kind of give you guys another overview here. This is another figure of what we just drew. All right, so check this out. This is on page 292. What you see here, this is your nerve, right? This is the nerve that I drew. This whole thing is the muscle fiber. Nervous impulse gets here. See these little bubbles? Those are called vesicles. They contain the neurotransmitter. Those bubbles are releasing acetylcholine onto the muscle fiber. But do you see that little just like white sheath that wraps around the muscle fiber? That's the sarcolemma. The sarcolemma gets stimulated and it sends an impulse all over the muscle at the same time. The sarcolemma then kind of interfaces with this little tube right here. You see that little tube? That's called a T-tubule. A T-tubule is just like a little highway that dives deep into the myofibrils of the muscle to get that electrical impulse deep into the muscle where all the sarcomeres are. Okay? When it gets down in here, you guys see this little blue region, that little blue stuff? That's the little bags of calcium. That's what holds the calcium. And when that electrical impulse reaches those little bags of calcium called the Sarcoplasmic Reticulum, that calcium gets released and the crossbridge cycling starts to happen. Now we can talk about rigor mortis, okay? So, when an animal dies, you still got plenty of ATP left over in the sarcomeres, right? The first thing that kind of falls apart is the Sarcoplasmic Reticulum. And that's this little blue area. Here's a blown up version of it right here. So again, these are little sarcomeres. This is the Sarcoplasmic Reticulum that contains all that calcium. This is the first thing that kind of falls apart. When it falls apart, what's going to get released? A bunch of calcium. Calcium gets released. Do we have ATP in the muscle cells, even though the animal's dead? Yeah, you've got quite a bit, right? So that's going to kick off crossbridge cycling, right? So the muscles are going to start contracting. It's eventually going to run out of ATP because the dead animal is not making new ATP. What's going to happen when ATP runs out? it's stiff because all those muscles are stuck in the contracted position. And it's going to stay that way for a couple hours. Rigor mortis ends when the proteins of the muscles start to decompose. And so finally, the actin and myosin kind of like fall apart. But that's what causes it, It all, it all kind of comes back together.
How is that whole process?
Oh, it's funny. It depends on it depends on the species of animal. I was doing this study on lizards, and we were looking at how strongly they could stick to different surfaces, geckos specifically. And our treatment was to explore how strongly the geckos could stick to a wall when they're alive and then after death. Okay, but we didn't want rigor mortis to happen because that would ruin our experiment. So I did all this morbid literature research into how long rigor mortis takes to kick in. And for like reptiles and most like ectothermic animals, you're looking at like 8 hours, like 6 to 8 hours or something like that. Humans, I think it's like 6 hours, 4 to 6 hours. And other like large mammals, right? A whole bunch of different factors affect it, such as how much exercise was being completed right before death and all that stuff. It's just like the worst morbid topic to do research on. But anyways. But that's what happened, okay? Good. And then see this, guys? I just wanted to show you guys, I'm going to zoom in here. This is just another example of troponin and tropomyosin. See all the binding sites? When the muscle's at rest, all those binding sites are covered by tropomyosin. But when calcium gets released onto the muscle, it binds to troponin, and it shifts that tropomyosin out of the way. Now all these guys are exposed, and myosin can grab on and start pulling. So that's a really good figure in the book. So it's on 292, and another good figure in the book that kind of summarizes the Crossbridge Cycling of the Myosin head is on the next page, 295, and this is really just what we just showed, right? Reaches out, grabs, ADP and P fall off, it pulls, it's stuck there until ATP comes along, it reaches back out, and it tries to grab if it can. Yeah?
Sorry, I just want to know, how does it feel like? Do you strangle them?
Oh, no, it's overdose of ketamine. Yeah. Which is actually like the easiest way to go. Easiest way to go for the poor geckos. So you give them an intercardial injection of ketamine, and it just suppresses everything. It was like the most humane way. What's that?
Do your coworkers know you're a killer?
Yeah, so you know what's funny about this? So we euthanized 5 animals, right? Which is just like nothing compared to all the research. So all of the research projects, even if you're just studying an animal for looking at its behavior, after the study's over, you have to euthanize all your animals. You can't give them to people because then it's like an invasive species, right? You can't release them back into the wild because then it's like an unnatural introduction. So yeah, all biological scientific studies, like a lot of animals get killed. All my fish studies, oh my gosh, it's just,
Wait,
you don't
even
know
if
what
you're
to
just
know.
like watching their behavior, like you're not changing anything you have to keep on?
Yeah, if it's in the lab, yeah. If you like get animals in, I mean, you can keep them around, but you can't just give them out to people because.
Could you keep them?
Yeah, you could keep them as pets. Yeah, we keep them around because I don't know. So anyways, and so the point is most scientific studies. I mean, you're looking at hundreds, thousands of animals that probably didn't make it, especially with like invertebrates and fish and stuff. We euthanized 5 animals, and we learned a lot from those animals.
What were they?
They were tokay geckos.
Oh,
Yeah. You guys are broke mean. They're like big, mean. They're like really large geckos. You can buy them as pets, but we studied them because they are really strong.
They're mean because they knew you.
With that,
Yeah.
they're like the worst pets because they're just very aggressive naturally.
Those aren't the ones in the keys.
No, I don't think
Those
so.
are ones.
I know, but they have.
You don't have to.
geckos
You
in
don't have to
the keys.
euthanize those. You kill at least 3 or 4 dead
They live. They're naturally. They live in like Southeast Asia, and their bite's so bad. I got bit so many times. Like their bite will give you stitches. It's like that bad. They have these horrible teeth. They'll bite and then they'll lock on, and then.
They're gonna kill you though.
That's how they warmed up to the
There was one locked onto my finger so bad I was in the vivarium for about half an hour, like Googling how to let them make blood's all coming out. It's just like, if you try to like, you know, squeeze their head, you could break their jaw before you can release them. It's horrible.
You had to break the jaw?
No, actually, because I didn't want to hurt them because we were using them for experiments. Finally, after like half an hour, I just startled him and he ran off. I had him dunked underwater and he was going to drown before he let go. And but anyways, the whole point of that story, was we published this study. We learned a lot about these animals, these 5 animals that got euthanized. And I got so much hate mail. I got people, you know, threatening to retaliate. You know, people were posting my advisors and I's home address on the internet. My advisor had to get like 24-hour police surveillance. And I was like, look, are you kidding me? Like, you know how many fish that have died because of all the other studies I've conducted? And no. Yeah. Anyways, so cool. Let's talk about. We're gonna stop talking here pretty soon. Let's talk now just about another study. Oh, you guys are gonna love this too. So, when I was teaching this physiology class at the University of California at Riverside, when I was finishing up my doctorate, I taught this great physiology class, and one of our labs was we took a bullfrog and we euthanized it. And the students themselves actually, like, PETA hated this lab, but it was a really great lab. I don't even think they can do it anymore because PETA tried to shut it down every year. But so you would take this bullfrog, you would euthanize him. The students would dissect out the gastrocnemius muscle, so live living muscle. Gastrocnemius is the calf muscle. It would dissect out that muscle. We would hook it to this apparatus that looked like this. It was pretty cool. So it was, so it was like this little stand. I don't want to draw before it's dry. So it was the stand, right? And it had a little hook in it. And what I would do is we would take the tendon of the gastrocnemius muscle, and here's the muscle itself, right? So you would hook it to that hook, and then you would take another hook and put it through here. And then this hook was attached to a little device that measured force. It's called a load cell. Right. Then we would take another little electrode that delivered a shock, and we would put that through the muscle. So what this allowed us to do is it allowed us to stimulate the muscle with an electrical shock. The muscle would contract because it's alive, and we could measure how strong the muscle contracted. Okay, and the results of this are really cool, and it lends to what we're talking about today. So what would happen is, and this is a plot of force as a function of time. When the muscle's just sitting there, it was like a kind of a small baseline force of, I don't know, the units don't really mean anything. We'll just say this is like 0 to 10 newtons. We'll say there's like a small baseline force. And what we would do is we would hit that muscle with a stimulus, like a little shock, that was maybe a volt. So like a 1 volt shock. When we hit that muscle with that stimulus, do you think that the muscle started contracting immediately, or do you think there was a little lag? It would take a couple milliseconds, okay? It would be like a millisecond delay. That delay is called the latent period. Okay, and that latent period is from like here to like right here when the force starts increasing. What causes that latent period? Yeah, like all that stuff takes a little bit of time. You know that electrical shock needs to go travel down the T tubules, the sarcolemma of the T tubules, sarcoplasmic reticulum needs to release some of that calcium. It takes a little bit of time, okay? And we can't get around that. So you get a latent period, then calcium gets released, and the force kind of looks like that. Force goes up, and then force goes down, okay? Force gets stronger, or what would make force get stronger in this little bump?
Shortening of the muscle, you said there's more strength available when it's mid.
Well, no, that's a great point. But in this case, the muscle is constrained at one length. So there's no change in the length. Think about calcium, right? At first, there might be a little bit of calcium being released, so you get a little bit of force, and then more calcium, then more calcium, and more calcium, until you get most of the calcium's being released. You got all that crossbridge cycling. Then all that calcium starts to get sequestered back into the sarcoplasmic reticulum, right? It gets pulled back out of the sarcomere back into the bag of calcium, okay? And that's why force goes down. Like calcium's being released and then being reuptaken, okay? That all happens naturally. Like every time we contract, calcium gets released, then we pick it back up. All right, now, after that, let's say we hit that muscle with like a 5-volt shock. So a stronger shock. Do you think the muscle will contract the same force, but for a longer period, greater force, but for about the same amount of time, or it's going to contract exactly the same?
Greater force, same amount of time.
Yes, perfect. That's exactly what would happen.
How many get those?
Do we still get the latent period?
Yes.
Is the latent period the same?
Same.
Yeah, it has to be, because that's just, we're constrained by that. So you get the same latent period, but now it's a bigger force. Why is that happening?
It's the same amount of time.
Yeah, about the same, maybe like a little bit longer, but about the same amount of time.
More electric pulses.
Well, volts is really the pressure, right?
Well, it's just the amount of like electrical,
Force.
yeah, it's the amount of electricity kind of going into the muscle. So if you have more electricity going in the muscle, what's it going to stimulate more of? More muscle fibers, okay? This is what's going on. So when you shock it with a little 1-volt pulse, it's only going to stimulate the muscle fibers that are like right beside the electrode. But when you stimulate it with a 5-volt signal, it's going to stimulate more and more fibers all throughout the muscle. It's going to spread out more. Yeah.
So no matter what the voltage is, you're always gonna have a latent period?
Yes. Because it just takes time for the calcium to be released.
Even if it's just like continuous? Or would
Well,
it be that way?
if it, no, we're going to talk about that here in a second. But if it's continuous, then something else happens. Yeah, it still needs to get released. But here's the deal. This is how you control the force, how much force your muscles produce. If I lift like a marker, I don't have to produce that much force. So I'm only sending an electrical impulse to like 1 or 2 muscle fibers in my bicep. Okay? If I want to lift something heavier like this book, I'm sending electrical impulses to like a lot more fibers in my bicep. Right? So I'm able to control the strength of contraction based on how many fibers are activated. Kind of makes sense to you guys? Just like this. Now, let's say you hit it with like a 2-volt signal, but it was in like pretty rapid succession. What would that force curve look like?
Be a longer period of time.
Yeah, so it would contract for longer for sure. Would force be higher, lower, less, same?
Not the same as 5. I don't think it'll be a little less than what the 5 was, but maybe the latent periods will be shorter.
Okay.
I'm thinking.
All right. This is tricky. So you'll get an initial force that's not as high as the 5-volt because it's like a 1 or a 2-volt signal. It'll go up. It'll start to go down, but before it can completely relax, it gets stimulated again. Goes up, 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 until it finally just levels out.
Is this like what happens in the EKG? Like, in the heart?
We'll talk about the heart. That's AP2. It's different.
Isn't it like a maximum amount of saturation the muscle's going to hit before? I'm curious.
Yeah. This is called complete tetanus. When it levels out, this is called tetanus. This is what's happening. First shock, you release calcium, right? Force goes up. Calcium starts to get sequestered. But not all of it gets sequestered. Second shock, what's going to happen? More calcium. Starts to get sucked back up. More calcium. Until finally all the calcium has flooded the sarcomere and there's no more to be released. You're not letting that calcium get sucked back up.